Class, let's look at the action of the secondary messenger system. Now, there are several secondary messenger systems. Remember what I said, it's kind of like a butler. You can't get in the house, so the guest comes, cannot get into the house, but you want to send a message to the owner of this mansion. Butler comes to the door, you knock on the door, you're the first messenger. The butler takes the information. He becomes the second messenger and takes it to the owner of the house. That's essentially how this works. Remember again that the water-soluble hormones cannot enter the cell because they can't get through the cell membrane. So the receptors are on the surface. So therefore, the receptors must turn on enzymatic cascade reactions that deliver the message to the intended intracellular source. All right, so here's one example, and this is called the cyclic AMP system. So hormone first messenger binds to receptor. There we are. Receptor activates something in the cell membrane called G protein. G protein activates an enzyme called adenylate cyclase. This enzyme converts ATP, adenosine triphosphate, to cyclic adenosine monophosphate. This is the second messenger. Okay? Then the second messenger activates some protein kinases. Now, a protein kinase is an enzyme, this is a generic term, that causes internalization, okay, energy to occur in another protein. So it takes the P off of ATP to energize another protein. So cyclic AMP activates protein kinases. The protein kinases then come in, here we are, protein kinases, and then this triggers the response within the cell. So let's go further. So cyclic AMP signaling mechanism activates kinase. They phosphorylate. In other words, what they do is take a P off of the energy compound ATP and activate other proteins, giving them energy. It's rapidly degraded by something called phosphodiesterase. So phosphodiesterase degrades cyclic AMP, and adenylate cyclase forms it from ATP. The intracellular cascade is one in which I alluded to, that is a geometric progression that the reaction becomes larger as it goes along. Amplification process. Okay. Because of this amplification process, here's something else. If this cell, let's say, had 100 receptors for this hormone, all you have to generally occupy are 50 receptors to get 100% of the action, which is different than the steroid hormones. Activate 50, let's say, I'm just using theoretically, let's say it was 100, it would be far more than that, I'm just saying that. Then you could activate half the receptors and get the full effect. Why? Because of this secondary messenger amplification effect. Okay, let's go a little further here. So you can look at the PowerPoint. This is a step-by-step. I'm not going to do that. You have the PowerPoint, so you can look at it. So now I'm going to another secondary messenger. Remember, cyclic AMP was just one. This other secondary messenger is called PIP, phosphoinositol phosphate. And it's a calcium signaling mechanism used by some amino acid-based hormones. Again, secondary messenger surface. Also involves G protein, and this one activates a phospholipase C enzyme. So this is a different one that we have. Okay, just naming some. Phospholipase splits membrane phospholipid, phosphoinositol phosphate, into two secondary messengers, diacylglycerol and inositol triphosphate. Diacylglycerol activates inositol triphosphate, triggers release of calcium. And so this is a calcium reaction. You can study it. This is a calcium, but this particular one is another secondary messenger as a different one for cyclic AMP. Different hormones in different cells use different secondary messengers, to say it away. Okay, so we go further. Now, the steroid hormones, on the other hand, the steroid hormones can pass through the cell membrane, and also thyroid hormone, even though it's an amino acid hormone, it can pass through also. And so what happens in this case is, since it can pass through the cell membrane, then it can diffuse into the target cell and bind with receptors inside. Sometimes the receptors in the cytoplasm, and sometimes they are directly on the DNA. But you form a receptor hormone complex that can enter the nucleus, but however, some can go straight in. But in this particular case, you would bind, and then you would go to a certain region of the DNA and turn on that region by turning on transcription factors. And then, you know the story, messenger RNA would be formed, protein synthesis, and that's how that particular one would work. Okay. Again, this is a series, and you can look at that yourself. Okay. Now, target cells must have specific receptors, which the hormone binds. So the target cell would have specific receptors for that particular hormone. Okay. Now, sometimes the receptors can have analogs, which means another substance can bind to the receptor, but there is specificity. The two things I'm going to say on hormone receptors are specificity and affinity. Affinity means binding power. Specificity and affinity. Target cells must have specific receptors which the hormone binds. ACTH, adrenocorticotrophic hormones, are only found on certain cells of the adrenal cortex. These are examples. Thyroxine receptors are found on nearly all cells because thyroid hormone can activate and work with many different cells. Okay? But this is a specific one. ACTH, adrenocorticotrophic hormone, which we'll talk about a little bit later. But this is specific. Thyroid hormone has many receptors on many different cells. Okay. Target activation depends on three factors. What's the blood level of the hormone? The higher the blood level of the hormone, obviously, the more receptors can be activated in the body. The relative number of receptors on the target cell. How many receptors? Is it 1,000? Is it 10,000? Is it 20,000? And remember, I told you specificity and affinity. How much drawing power does the receptor have for the hormone? How much drawing power? How much binding power and drawing power? Okay. So we go back and we say that hormones influence the number of their receptors. Okay, here we go with something called upregulation and downregulation. Okay, let me kind of give an example. Let's say the hormone level in the blood is low, but the target cell wants to really use it. So it wants to capture as much hormone as it can. So what it will do is build more receptors. In certain cases, the receptors, like for surface receptors, are in the cytoplasm, and upregulation would merely mean to take some of the receptors in the cytoplasm and put them on the cell membrane surface. Now, what would that do? That then would allow a low concentration hormone to activate more of the target because you're putting more receptors out there. And the more receptors you activate, the more the action will be. Downregulation would be this, that the blood level of a hormone is too high. The target cell does not want to activate as much as the blood level is dictating. So the thing is to take the receptors in. All right, what's an example of this? Let's say you had an antenna signal, an antenna, and there was a weak signal coming from the TV station or whatever. This is old days, but a weak signal. Then the person would either raise the antenna higher or put 2 or 3 antennas there. That would capture the weaker signal. If the signal was too strong, then you'd pull the antenna down or reduce the number of antennas using kind of the old day way with the antennas on the top of the house, unlike the cable today. But that gives you an idea of what this up regulation and down regulation are. Okay, so we go a little further. Hormones in the blood. Okay, hormones circulate in the blood either free or bound. Now let's explain this. The blood is made up of a lot of water. Okay, lipid soluble substances don't travel good in water in the blood. So most lipid soluble substances must have a carrier protein, something that can surround them. The carrier protein would be amphipathic, which basically means it's part lipid soluble and part water soluble. But these act as carrier proteins. Like, for example, for thyroid hormone, you got thyroid binding globulin. And you have various carrier proteins, okay, that carry it. Water-soluble hormones don't need this because they're already water-soluble, so they can float nicely in the blood. Now, when it's a lipid-soluble, like a steroid hormone and so forth, or thyroid hormone, there are 2 fractions in the blood. One is a free fraction, and the other is a bound fraction. A free fraction and a bound fraction. Now understand, they are in equilibrium. So other words, when the hormone is in this binding state, then when it gets into capillaries, we talked about blood vessels, where hormone is released, then some of the hormone the immediate hormone that would be released is from the free fraction, because it's merely being carried in the blood. It's not dissolved that well, but it's a little bit free. That's the most active. Now, when that exits through the capillary going to the target cell, then that opens up some space. Let's say in the fluid of the blood, to take some of the bound and replace the free. So they're always staying in equilibrium. Now, if the bound is gone, what happens? The source cell that made the hormone will replace the bound fraction, and that will cause equilibrium again. So again, the free fraction is the most active. That's what you're working from. You can look this up even more. So the concentration of a circulating hormone reflects its rate of release. The faster the release, now there's 2 things. One thing is, what's the source cell? The source cell can increase the concentration, and the area of the target cell can reduce it because it's leaving the circulation. Another thing is the speed of inactivation. Hormones are inactivated by enzymatic activity. So some hormones are inactivated, they're removed that way. Some are urinated out, haven't been degraded. So again, there are various factors involved in the concentration of a circulating hormone. Okay. And as I alluded, hormones are removed through degrading enzymes or the kidneys or liver enzymes. Liver is the main thing for degrading things. Half-life means if you had 100 milligrams of a hormone, let's say 100 microunits, how long would it be before it's degraded to 50 micrograms? I'm going to close right here and pick this up on the next one.